My name is Rachel Dockman, and today I want to talk about the establishment and plasticity of the rumen microbiome. I became interested in this topic after talking with Dr. Garrett Swen from Wisconsin about his research. Dr. Swen, during our discussion, raised an interesting question. How do these calves get colonized by such a unique assortment of bacteria? Where are they coming from, and is there any way that we can change it to impact their production later on in life? These questions really intrigued me, so I really wanted to learn more about what's going on to lead to calves becoming colonized the way they are, and what can we do to change this? So the microbiome is a pretty hot topic in nutritional sciences, the agricultural sciences, and health sciences, pretty much all sciences today. We know that the microbiome has about 100 times more genes than the host. And we're seeing lots of importance of this topic in livestock and in humans. In humans, it's been linked to different gastrointestinal diseases such as Crohn's or irritable bowel syndrome. It has been linked to obesity. There are some species of bacteria, such as a fecal bacterium, that is a higher amount of it's linked to a greater chance of becoming obese in life. There are also some neurological conditions in humans that, with more and more research, we are finding to be linked to the microbiome. For example, Parkinson's or the autism spectrum disorders. They're starting to see that change in the microbiome of people suffering from this results in positive changes in behaviors and in physiological outcomes. In cattle, the microbiome is extremely important in dairy production, the health of calves, feed efficiency. Cows get roughly 90% or more of their energy from the rumen, which is colonized by microbes that ferment their food and break it down. So it's obviously a huge part of the health of cattle. To really understand the ruminant microbiome, it's important to know how it's being established. There are three primary factors that come into play: the microbiome itself and the bacteria and microbes that make it up, the genetics of the host, and the environment that's in. The environment can include feed that the cow is being fed. For example, high starch foods tend to lead to more lactic acid producing bacteria, which can lead to physiological changes in the host, which will lead to differences in the microbiome, but that's more production. With establishment, it's important because the microbes that are present in the environment will be the first ones to enter the calf when it's being born. And the genetics that a calf has may predispose it to a greater diversity or a specific subset of microbes that other ones may not be predisposed to. And then the different microbes that are colonizing it will fight with each other and compete. So there are many different factors at play. Initially, however, the calf colonization is pretty straightforward. Most of them get their microbes from about the same things. When they're first born, the mother will lick her calf and they'll get some of her oral microbiota. They'll get some of when she is ruminating and she's bringing on bolus of the rumen, so they'll get some of her rumen microbiota. And there's also when they suckle, there are certain bacteria that are present in the milk or they're present around the teats. So when the calf is suckling, it'll get some of those in its system. Then there's also, during birth itself, there are many different bacteria, especially methanogens, are found in the vaginal canal. And there's also some exposure to feces and fecal matter, which can contain lots of E. coli, which is actually a pretty large problem with some cattle because they will be predisposed or exposed to this and can develop scours or diarrhea, which can be pretty dangerous. There's also been some recent research lately looking at an actual placental transfer of microbes. Previously, it was pretty much thought that the placenta is sterile, that there's nothing in there, the calf is 100% germ-free, but they've actually been doing some studies where within 20 minutes of birth, before the mother can touch it, they opened up the calf, they euthanized and opened up the calf, and they found microbes in the rumen contents, in the cecum, fecal matter, and in just throughout the gut. So there is some evidence that some of these are coming from the mother, and they do actually represent the oral microbiota more. And these are calves that were never licked. So this shows that there might be from what she's eating going through her bloodstream to the placenta. So colonization is coming from many different places. Now across the board with different calves, they are going to have a pretty similar microbiome simply because they are drinking milk. So they're going to have these bacteria such as lactobacillus or bifidobacterium that you really will not find in adult cattle. and they tend to have more of a lots of different functions in what their bacteria do. They have a less diverse population, but they fill different niches because they're not really colonizing the rumen as much yet. There are some fibrolytic and methanogens found in the rumen, but they're not eating solid food when they're young. So they don't have these specific niches with tons of different microbes like adults do. And due to how much they're growing, since their diet's changing, they tend to start eating dry or solid food at 4 weeks old. So since there's so much change, it's a very fluctuating microbiome, and it's hard to look at specific genetic influences in young cattle. So you don't usually start seeing these genetic influences until they're about 2 years old. Adults, they're just so heavily dependent on the feed. So different feed will result in different outcomes for the adult. And they don't just have bacteria, they also have protozoa and fungi. Now, less is known about the colonization and these 3 major factors, but there is more research done in adult cattle. They've looked at genetics, such as looking at breed influence and how feeding them different types of feed will change or help them keep more stable microbiomes, or how the microbiomes may be just more stable in general than you'd see a calf. So you are able to track a little bit better the production ability and how it's being impacted in adults than you are able to in cows. Lots of research is really going into which is having a bigger influence. Is it the genetics of the host, or is it the microbes that are colonizing it? It seems like they're kind of influencing each other. So the genetics can influence what's colonizing it, but these microbes might influence the way that genes are being regulated. It could influence inflammatory factors and immune responses, which could kill off some microbes, keep others around. So there's this big debate on which one is kind of easier to overcome, because when you find the easier one to overcome, you can help get more production in more efficiency in these animals, which is always the goal in livestock production. When it comes to the genetics in the microbiome, as I said before, it's hard to really measure the influence of genetics with calves just because there's so much fluctuation, but there are some links being made between breed and their microbe populations. So there's one study where they had Angus and Charolais, as well as some hybrid hybrid sires, and then they measured the efficiency of their calves later on in life. And they actually did see that Angus, they tended to produce cattle that had a more stable microbiome. So even when they were switched to a low efficiency diet, they were still keeping up a pretty efficient microbiome and doing good fermentation parameters, and showed some pretty good results. So this is showing that maybe the genetics, even though the microbiome is being influenced kind of by this food, genetics might be winning out a little bit. My favorite method that is being used in the recent years, I think the first study I saw was from 2010. They've been doing them, though, pretty consistently, is a rumen content transfer. So they actually cannulate 2 cows or cattle, and they will take the rumen contents out of one and put it into the other. And it's been showing some pretty fascinating results. They've done this with dairy cattle, where they actually transfer the rumen contents from a high-producing cow into a low-producing cow, and they did see an influence. The low-producing cow produced more milk briefly, but then very quickly the volatile fatty acid concentration, the pH, a lot of these parameters within a day went about the same to how they were before. Now the bacteria actually took a lot longer. It took 2 weeks or more for the bacterial population to become about the same. But even then, it didn't become exactly the same. There were differences. In a study with beef cattle and their fermentation parameters, their fermentation ability was about the same, but there were actually some phylotypes that once they switched contents, they didn't see them anymore. They had previously seen them in these steers, but they were gone. So there is some evidence that you can make some long-term changes by just taking out all traces of a specific microbe and replacing it with a new one. And this has a lot of potential for changing the way that we can control what an animal is able to produce without necessarily relying on breeding and genetics. So livestock people in particular, but also human researchers have really been looking into this, and a lot of research has been coming, and there is a huge future for microbiome research. And there is contributions it'll be making towards human health as well as livestock efficiency. It's vital to understand this for livestock, because if we can understand how that early colonization is impacting the population of the microbiome later on in life, we can determine how genes are actually linked and how they're influencing this, and how we can manipulate this microbiome. We can change what the cow is ultimately producing. We can help them to develop more efficient microbiota. We can give them probiotics if we find it more efficient to control the actual bacterial populations, or we can breed for certain genes that are going to lead to better microbes. We'll be able to ultimately reduce the methane emissions, the carbon footprint, which are all huge issues in livestock production, and use less land and resources to produce the same amount of these products. My favorite topic personally is more looking at it with human health. And I don't have much more time, but I just think it's really impressive what we're going to be able to do with this in the future and what we're already doing. We're going to be able to discover new ways to treat these disorders that are being linked to this, such as, autism disorder. That is showing a huge link. And if we can just work on a simple probiotic, a simple antibiotic that is going to help with these microbe populations, this could be an amazing treatment and provide awesome opportunities for people who have not had these before. We can see more about how the environment is affecting us and what we can do to take our own health into our own hands. So there's just a massive future for this. So thank you so much for listening to me, and I hope you enjoyed it. Thanks.